The overall goal of this protocol is to quantitatively measure arenavirus particle attachment to host cells.
The main advantage of this technique is its high sensitivity. It can also be adapted to measure virion endocytosis, as well as the release of genome into the cytoplasm following fusion.
To begin, to make complete DMEM, to a 500 milliliter bottle of DMEM, add 50 milliliters of heat-inactivated fetal bovine serum and 5 milliliters each of 100x penicillin streptomycin and 100x HEAPS buffer. At the end of the day, seed 2.5 times 10 to the 4th Vero E6 cells per well in triplicate for each virus in a 48-well tissue culture plate in a final volume of 500 microliters of complete DMEM. Incubate the plate at 37 degrees Celsius in a humidified incubator with 5% carbon dioxide for 10 to 18 hours. Under a Class II biosafety cabinet, using ice-cold complete DMEM, dilute the Hunin Candid 1 virus samples to be tested to 200 to 2,000 focus-forming units or plaque-forming units. Plaque-forming units will be referred to as PFUs in the remainder of the protocol. For example, to inoculate with 50 microliters of 2,000 PFUs in duplicate, add 4,800 PFUs into a total volume of 120 microliters of medium. Store on ice until ready to add to the Vero cultures. Remove the plates from the 37 degrees Celsius incubator and place on ice or at 4 degrees Celsius for 15 minutes to inhibit the ability